Okay. Well, first of all, thank you for inviting me. It's a tough act to follow, Don, but I'm going to do my best. And I'm going to try and pick up on a few things that Don has picked up on as well that I think link in to what we're trying to do on the other side of the pond in Europe. As the title suggests, I'm going to talk about two things. One is an attempt to try and coordinate ME research across Europe, and the other is to focus on what we're trying to do in the UK, and in particular at the institute that I'm a member of, which is the Quadram Institute, that used to be called the Institute of Food Research. So I'm going to start by really sort of highlighting this, I think. So why haven't we really made any significant advancements to date in understanding the cause of ME? And I think what's here you're all aware of. You know, we don't have enough people on our gut, and it works both ways. Now, an important part of what's in your gut are microbes, and there are lots and lots of microbes that live in your gut. In fact, they populate all the surfaces of the body, but by far the largest number are found in your gut. And just to give you some idea, there are about 2 to 3 microbes for every cell that we have in our body. So, I mean, they outnumber us significantly. There are 1,000 to 10,000 different types, what we call species of bacteria. Their genome is massive. They're 500 gigabytes, which is about 100 times the size of our own genome. And these encode over 3 million proteins. And our genome encodes maybe 100,000 proteins. So you can see the microbes in our gut not only outnumber us, but they can also make many more things than our body can make. But normally we live in harmony with our microbes, and they're actually essential for our health. And what's interesting is that by far the most prominent population of microbes are viruses. So viruses outnumber bacteria, and in turn we have fungi as well in there, protozoa and archaea. And if you think back to one of Don's slides and these infections that can be triggers for the disease, all of the components live in your gut. So the interesting question here is, do the infections arise from within rather from outside the body that could trigger the disease? So because of principally sequencing now, our technology enabled to sequencing genomes, identify microbes based on genetic fingerprints, we've got a pretty good idea about what's there. And we know that over our life course, they undergo quite striking changes. So we're essentially born sterile, but we get rapidly populated with microbes. And then as we age and our diets change, the microbial populations change as well. And as we approach old age, again, the microbes change. And it's in parallel with changes in our physiology, things we eat, things we get exposed to in the environment, where we live, how we live, our behavior. All of these are impacting on this process of aging and changing our microbes. However, what we know is that we all have a core. We all share a core microbes. So there are certain types of microbes that all of us have, about 57 different types or species that most of us have. And there are principally 2 major types or groups of organisms there. And not surprisingly, this core provides essential functionality to the body. They're involved in digestion principally, so they help us digest our food. So without the microbes in our gut, we wouldn't be able to get the maximum benefit from vegetables and plant material that we consume in our diet. They're essential for us to be able to digest this. They also produce other things like fatty acids, and that's a source of energy for the body. So our microbial metabolism can contribute 15 to 20% of our daily energy needs. So they're an important source of energy and nutrients to keep us healthy. And they also produce a variety of amino acids and vitamins, again, things we can't get from our diet that we rely on our microbes to keep us healthy. So they're an essential part of keeping us healthy. But there are cases where they can also contribute to disease, and this really comes about through different interactions. So our own genes, our genome determines the type of microbes that populate our body, and then our environment, where we live, how we live, what we eat, what we get exposed to, can also shape our microbes. All of these are important in establishing the populations that live in our gut. But they're susceptible to changes through changes in our diet, stress, drugs we might take, toxins, and they can cause a change in our microbes. It's called dysbiosis or abnormality. So the populations change. Some die, some bloom and grow and outgrow. And these can cause leaky barrier in the gut, and they're allowing them to get into the body where they can stimulate a reaction, an immune response, which can cause disease. A good example of this is Clostridium difficile. So this is a very difficult infection to treat, but it arises because generally we're taking too many antibiotics, particularly in hospital. We're killing off beneficial microbes. Clostridium difficile can survive those antibiotics, and it can grow and increase and start to produce toxins, which can cause a leaky barrier. And it's very difficult to treat. Okay, but also these other diseases here are associated with this change in the populations or the activity of microbes in our gut. Now, whether or not they're a cause and effect is an interesting question, and for some, the evidence is stronger in favour of a cause rather than a consequence of the disease. But interestingly, ME could be one of the diseases that has an altered gut microbiome. And so not surprisingly, there have been a few studies. They're accumulating now. There may be around 10 now. But the problem is, and as Don's related this earlier, it's very difficult to compare across studies because they're using different patients, they're using different diagnostic criteria, they're looking at patients of different severities, and the methods they're using to analyse their gut microbes are different. So it's difficult to compare across studies. But what's clear here, and don't worry too much about the names, it's clear that some microbe populations go up and some go down. So there's clearly a change in gut microbe populations in patients with ME. Okay, so there's clearly some alterations. What we're interested in Norwich is looking at the viruses. And I told you the viruses by far outnumber the microbes. And viruses can actually account for some of these changes in the bacteria that I've just described, as you'll see. Now, viruses don't come in just one flavour. They come in a variety of types because they infect different types of cells in your gut. So we have viruses that are very good at infecting bacteria. There are viruses that will infect our cells in our body. And there are also viruses that will infect other types of organisms in the gut called archaea. And then there are these things called bits and pieces of viruses that can actually change the behaviour. So it's a complex mixture of viruses that live in our gut. And as I said, there are maybe 10 to 20 virus particles for every cell in our body. So they vastly outnumber us. There are, again, not surprisingly, the core. So there's about 20 to 30 types of viruses, and it's these types of viruses here that are in most of us. Okay? But what's interesting is that they only account for about 4% of the total population of viruses in the gut. So we're looking at a very small proportion of the viruses. And we know they can influence the microbiome. We know they can change their activity. They can kill bacterial cells, causing disturbances. They can infect the cells in our body, which can cause disease. And they're highly individualized. So everybody has a unique repertoire of viruses and bacteria. And we now know from very recent studies that the virome in the gut, the virus in the gut, are changed and linked to gut diseases, particularly inflammatory bowel disease. So there's increasing evidence that links them to causing diseases in humans. This is just a picture of, this is a single bacterial cell, and these are the viruses that are infecting this bacterial cell. So you can see each bacterial cell has many, many viruses attached to it or that actually invade that bacteria. And these are the types of viruses. These are the very characteristic shapes. Here we've got ones that sort of look like sticky balls, and then these here sort of like alien spaceships. So what they do is, you can see they attach to the bacterial cell through these structures here, and then they inject their DNA or toxins that are in the head directly into the cell. So, and they can kill the bacterial cell, or they can just manipulate the activity of the bacterial cell. Okay, so this is not really new science. We've known about these these viruses that infect bacteria, which are called phages. They've been described many times. In fact, they've been used therapeutically to kill off bacteria that cause diseases. So in Russia, they're still being used now to treat bacterial wound infections and kill off the bacteria that can cause sepsis. So the potential for using phage to treat GI diseases as well. And as you can see, in the USA, they're used to decontaminate food products, to kill off bacteria that can cause food spoilage. So phage therapy is not new, but I think we're looking at potentially a new application here for gut diseases. And so I've said that bacteria can kill cells, so that's called lysis. They can also infect but hide inside a bacterial cell. This is called lysogeny. And through these processes, they can transfer their genes from the virus to the bacteria. And some of these can give new properties to the bacteria, so they suddenly become resistant to drugs, which is bad news if this is a bacteria that can cause infections. They can give this cell an advantage in terms of competing with all the other bacteria in the gut. And they can also establish themselves and make sure the bacteria survive in the gut. And then the viruses that hide in the bacterial cell can, under the right conditions, kill the cell and start to release many more viruses to infect other cells. So most of the viruses in our gut are of this type. They're hiding within our bacterial cells. But if we change our diet, take a course of antibiotics, they can kill the bacterial cell, produce many more viruses, and the cycle of infection goes on and on. And if this is severe, then that has serious consequences for our health, can lead to inflammation and disease. And as I said, there's good evidence that these viruses are changed in inflammatory bowel disease, particularly these type of viruses. These expand in number in patients with inflammatory bowel disease. And this is seen in American populations as well as UK populations, so it's not just restricted to one type. And what's interesting is that in the study we've done with Maureen Hansen, whose group's at Cornell University in America, we've got some preliminary evidence that there are changes in the gut virome in ME patients as well. And so that's really the questions that we're interested in asking in Norwich. Do gut viruses play a role in the development of ME? And can we identify specific types of viruses that may actually cause the disease? So it's a team effort, and we're assembling over the period of time that we've been working in this groups of collaborators, and you'll notice that we are interacting with other groups in Europe. Jonas Bergqvist in Sweden. These are students and medical students that are actively involved in the research, and then we have consultants at hospitals that are helping us with patient recruitment, and then we have specialists who are very good at helping us analyze all the sequence data that we're getting from our patient samples to allow us to identify the viruses. So it really is a team effort, and it's taken several years to assemble this team to actually begin the process. This is the sort of snapshot of how and where we're recruiting our patients from. We've got two sites. One is southwest London, where one of the major ME centres is based at St Helier Hospital. And then this is where we are in Norwich here, and we're trying to recruit patients from Norfolk and Suffolk. You probably won't be surprised to know that both in these regions, there's not clear indication of how prevalent the disease is. An estimate is that with maybe 1,000 patients with ME in this part of the country, and maybe 3,000 here, but there's no accurate records. So this is one of the issues I think Don's also mentioned, is that we don't know just how prevalent it is. And this probably relates to issues with diagnosis and getting an accurate diagnosis. So we've decided to focus on severely affected patients. So we are having to make house visits to recruit patients and collect samples. And to date, this has now increased. We're now up to about 25 patients. I mean, Don as well has identified the issues that we have in terms of recruiting these patients. And I thought I would put this up because this is typical of the type of patient that we've been able to recruit to our study. So this is an individual that's now 38 years of age, but she suffered from the disease since she was 15. Before she was taken down with the disorder, she was very bright, very active, wanted to teach, clearly had lots of ambitions. But then she spent the last 18 years bedbound in a darkened room. She can't watch TV or read books. She can't have a bath. She can't wash. And communication, interaction with others is severely constrained by the disorder. So our main way of communicating is via text messages. And she has trouble eating and drinking. And a treat for her is when she's been able to eat a piece of cheese. So this is really a typical example of the type of patient we're really trying to encourage to get involved in our research. And the most encouraging thing with all of our patients is right here at the better, is that they all have a positive outcome, they have a perspective on life, they want to get better, they want to get involved, and they all hope for a better future, which is really amazing considering the condition that they're in. So this is what we do. We're recruiting patients. We're also recruiting healthy individuals that live in the same house. So we're trying to get healthy controls that are exposed to the same environmental factors, eat the same foods, etc. So we have 15 female severe ME patients. We have no males. All the severe patients we're able to recruit all females. And we have, from 15 of those households, 11 of their carers, spouses, parents have agreed to recruit. So we take a stool sample as well as a blood sample if we can obtain a blood sample. And this just gives you an idea of the processing of that sample. So we're interested in the bacteria, the fungus, and the viruses, and we're developing protocols to separate those different populations from the stool. We're also collecting sort of liquid phase of the stool to look at the metabolites, which is sort of the functionality, gives us clues as to what all these microbes are doing. And we use a variety of methodologies then to identify what's present in terms of bacteria, fungus, fungi, and viruses. And we're looking at DNA viruses and both RNA viruses. So the challenge is this sort of, I put this up here because this highlights the difficulty we have in attempting to do virus studies in the gut. So in terms of looking at viruses, there's no conserved gene that allows us to identify all viruses. They're all unique, all have individual genomes, so there's no sort of easy means of identifying them. They're very diverse, so they're rapidly changing and evolving. And in terms of isolation from the feces, there's really no methodology. So we've had to invent the methods to be able to do this. There are small amounts that we can recover, and so we have to try and amplify the sequences so that we can identify them. And then the issues with the sequencing, we've got to optimize all of these protocols to be able to do this analysis. And then there's also, in terms of all these sequences, well, how do you analyze it and how do you use it to identify what's there? Well, we're struggling because there are a few reference databases, so we're on a voyage of virus discovery. We're discovering viruses that nobody's ever described before, as well as those that people have described before. And so we're having to invent a lot of processes, and this is taking a lot of time to do. And this is one individual in my group, it's one of the PhD students that's supported by the InvestME charity, and his role is really to develop the protocols for isolating viruses from these samples. And so he's just finished optimizing this, so we're now starting to apply these protocols to patient samples. This is another PhD student, this is Fiona, who's been developing the bioinformatics to be able to take all the sequence data and develop the systems to be able to identify what the sequences belong to, which viruses are present. And so she is working with collaborators at Imperial College London, as well as the University of East Anglia to do that. And so as you see, we're building this team of different expertise to tackle this. And the end product is that we'll essentially have a publicly available, easy-to-use bioinformatic tool that anybody can use to be able to identify viruses in stool samples from different patients, different disease settings. So we're hoping to provide a valuable resource for investigators in the future. This is just sort of what we've looked at so far. This was our sort of proof of principle that we can apply some of this methodology. So really, all I want is really the bottom line here is that we can identify viruses that seem to be unique to our patients that we can't find or are less abundant in our healthy controls, which is encouraging in terms of why there are differences, and this may actually provide us with important clues. And these are actually some of the viruses that we've been able to isolate from the patient samples, these images here taken with an electron micrograph. So this is very encouraging. This was actually done for mild moderate, so patients are now working with the very severe samples. So this is, as I said, what we're doing next. Again, 2 other PhD students are actively involved in here. Characterizing the virome of severely affected ME patients. Do the viruses kill gut bacteria. So I said one of the causes of change in bacterial populations is viruses kill them. Do ME patients have viruses that kill bacteria? And could these viruses cause unexplained disease? And we're trying to do some more sort of investigative studies, taking the viruses and see if we can cause any symptoms in experimental animals if we transfer these viruses to them. And then, importantly, from an autoimmune response aspect is, do patients generate antibodies to these viruses as they would in an autoimmune disease? So we're testing the immune responses of ME patients to whether or not they have antibodies that react to their own gut viruses or bacteria, which would be an indication of an autoimmune disease that's triggered by this reactivity to gut pathogens. I'm particularly interested in this type of antibody, IgA. IgA is the major antibody that's secreted into the gut. Our immune system produces grams and grams of this antibody, and it's primarily there to neutralize, bind, and eliminate toxins, pathogens that live in the gut, to stop them invading and infecting. So it usually excludes things from getting into the body. But in diseases like inflammatory bowel disease, where you've got a leaky gut, then this becomes an ineffective defense because microbes can escape and enter into the gut. And what the body tries to do is produce even more antibody to try and neutralize it. So you can detect differences in the types of bacteria that these IgA antibodies bind to in a healthy individual versus a patient. So we've been looking at ME patients to see what their IgA profile looks like in the gut. And so what's interesting is this is results from inflammatory bowel disease patients, and this is the level, the proportion of their gut microbes that are coated with IgA. Healthy control, you see it's around 20%, but in patients with Crohn's disease, ulcerative colitis, this increases, indicative of an active immune response that's been generated to microbes that are in the gut of these patients. And in our analysis so far, what you can see is that there seems to be a trend that in ME, there's also increased levels of IgA antibody that are reacting to the gut microbes. And this is seen in this group of patients. We've got more as well there. So the trend is the same as we see in inflammatory bowel disease, which is interesting. As I said, we're interested in the liquid phase of the stool, looking at what the microbes produce. So this is metabolites, principally, that come about from the digestion of the food we eat. And as Don has illustrated, that in looking at patient's serum, there are changes in metabolites. So we're looking at what the gut microbes are producing. And so we've been comparing these fecal water samples from our severe patients and the people looking after them. And what we found is no significant differences in about 50 metabolites, which are listed. Some of them listed here. But what is interesting is this one at the bottom here. We've identified increased levels of metabolites from L-carnitine, which is usually present in meat. What's interesting is that some of these metabolites are known to have adverse effects on the brain, and particularly on the blood-brain barrier, which is made up of endothelial cells, which normally is tight and prevents things getting into the brain that could stimulate inflammation. Well, some of the metabolites here are known to make that barrier leaky, which could explain why there's some inflammation in certain diseases when these metabolites are high. So this is very preliminary, but it's an interesting observation that we're trying to follow up. Again, it sort of links the gut and the brain quite nicely. So now to finish off by sort of talking about what we're trying to do collaboratively across Europe. And this really comes about, again, the charity invested in me has been very keen in trying to develop this. And some of these issues, again, have been raised by Don already, so I won't really go over them. You know, in order to really make an impact with the science we're doing, you know, we need long-term commitments to fund the research. We need to build collaborations and teams of people that are focused in different centres. We need to also produce the science and not remember that the science needs to be for a purpose, diagnosis, prevention, solutions, and to instruct and inform policymakers as well to ensure that this is sustainable. So this is what I want to talk about, is how we're trying to accelerate ME research in Europe. And there are two relatively new initiatives. One is Euromene, which is funded by the EU, And this was to connect and establish a network of ME research. So it's to try and bring people together to start to engage and talk to each other and form collaborations. And then with the help of the charity, what we've formed is Emerge, which is to try and advance the research. So this organisation brings people together, and what we're trying to do here is to get them actually to work together on core, common research priorities across Europe. So this is Euromene, and as I said, it was to create an integrated network. It's around 6 million euros, so it's not an awful lot of money, but considering it's not to fund researchers, bring people together, that's probably sufficient. 15 countries, and this is it was initiated by Uldis Berkis, who's in Latvia, but it involves people in various European countries. And these are some of the activities that they're engaging in. So you can see it's really trying to develop standards for clinical diagnosis, establish communication links, foster knowledge exchange, review existing facilities, trying to establish biobanks that can be used for research. And then this is Emerge. So again, these are the countries that we're involved in. It's not just Europe. We are engaging with Americans and people in Australia, New Zealand as well. And this is to try and develop a common research agenda. And we've identified 3 areas that we're trying to focus on. One is diagnosis, okay, patient stratification. Again, Don has mentioned this importance of stratifying patients to try and almost get to personalized medicine, develop individual therapies, interventions. Biomarkers, trying to identify what are the effective biomarkers, what can be useful. Sample standardization. Again, when we get samples, we've got to make sure they're treated, handled, and processed in exactly the same way, so we compare results across different studies. So that's what we're trying to sort of work around. And we're trying to bring the 2 organizations together. We have cross-membership, so we have people actively involved in Euromene and the same individuals involved in Emerge to try and bring all these people together and get everybody to work together. That's really the objective there. And we are trying to look beyond Europe. So this is an important initiative by the NIH, Dr. Vicky Whitmore in particular from the National Institutes of Neurological Disorders and Stroke. And in working with the CDC, they were trying to develop a commons data element which could be used to guide studies that were undertaken, funded studies. So, and there were various working groups that were formed to look at different aspects of ME. And some of us have been involved in these groups. I'm involved in looking at the biomarkers group and immune markers. And again, it is trying to ensure that when people do studies in ME, that we can cross-reference and compare and contrast the results from different studies, because we're all working to a common data element. So that's very important, I think. And so I'll stop there. Thank you very much. I'm sorry if I've run over time.
Thanks a lot, Mr. Sean Kynning, for your interesting speech. Við ætlum núna að vera með nokkrar spurningar utan á sal til þessar ágætu fræðimanna og lækna. Og ef það eru einhverjar spurningar á borðum þá endilega skulum við reyna að safna þeim saman. Eru einhverjar spurningar komnar í hús? Við getum líka haft þann háttinn á að fólk rétti upp hönd og kynni sig og beri fram spurninguna, ýmist á ensku eða íslensku, og við reynum að þýða það yfir á enskuna með hjálp félaga okkar í ME-félaginu á Íslandi sem kunna kannski réttu orðin, ólíkt mér þegar kemur að þessum sjúkdómi. But I would like to invite Mr. Peterson and